Welcome everyone. This is the first tutorial from the cycle of tutorials that we are organizing. So the idea is to discuss how to model like the different frameworks and methods to model microbial ecology, with the idea of doing it at multiple scales. So from the genes all the way to communities. And the goal of these tutorials are three. So first of all, so that we can have like a deeper understanding of the model that is going to be presented, um, have an opportunity to talk to a person that developed and worked on it, and also to be a first level entry for those that want to start using similar or related frameworks. So there's always this question of how you start or from where you start. So this would be like a first level entry. And finally, so that we can make kind of a crosstalk. So the idea is that we have people from different areas, so from these different levels, um, with different modeling expertises, and we have rare opportunities to really like crosstalk and see how our models are related or could be combined in some way. So this is the general idea, and we'll try to have these talks like about once a month, between once a month and once every 2 months. And they will generally have this format where we have a speaker that talks about, gives a general overview of a modeling framework, and then talks about her own work. And then we have like a practical where we get some hands-on experience with the model. And then finally, we'll have a final minute to ask final questions and reflect about like how the models combine with other models or with our own work. Yeah, and I think a very good place to start are the consumer resource models that are very like used, very popular in ecology. And it's something that I personally have, I really want to learn more about it. And it's a great opportunity for us to hear Veronika that's going to explain all about it to us. Yeah, do you guys want to say something else?
No, it's fine. Let's start.
Okay, so to those who don't know me, I'm Veronika. I'm a senior PhD student in Sergei Maslov's lab. And I mostly work on different consumer resource of semi-complex microbial communities. And for the first tutorial, as I got this honor, I think I'm going to mostly describe you the core assumptions for those models and like most popular ones, because like what I'm working on is mostly like extensions of those models. So I don't think it makes a lot of sense to me to describe you our models from the start. So let's just start with a general intro. So this is going to be an outline of my talk. I'm just going to give you some general idea why we want to use consumer resource models, and then describe you like three major assumptions that are out there on the market right now. And then there will be some concluding remarks and like perspective for future work. And if you have any questions during my talk, please ask them in the chat, or you can raise a hand and maybe one of the organizers will just pop up and tell me that you have a question and we will clear it out. So to start with, at least my general motivation to dive into those community modeling was data-driven, because I was working with all this metagenomic and amplicon sequencing data, and like there was a surge of these data sets in the last decade, and people actually realized that microbial communities are very ubiquitous, they're very diverse, and we generally don't have much understanding why they're different in different environments, why there can be some blooms in some communities, why some species can die out, and like what is the stability of a state, like how to understand that a certain time point that you collected is stable or not. And also there are like lots of practical questions, like what if you want to study some community in the lab, how do you make sure that you will have like reproducibility of your experiments? Because like in a lot of cases, you just pick a sample from the soil or from human gut, and then like you have some random stochastic event that ends up in like various species compositions, and you can just reproduce whatever you wanted to do. And models are actually a really good instrument to address all those questions that arise from the data, because you can dissect and separately study different factors that may affect community properties. And also in the future, when you will develop a good model that actually describes your community property, you can use it to explore different experimental scenarios before you actually do an experiment, and then pick the most interesting identity and check it in experiment, and thus you will save money and you kind of will optimize your research. And also, like as a far, far future, what we hope to have in all those models is that if we have a predictive and descriptive models, we can actually use them to test and design different strategies to engineer, manipulate microbial communities, and like do all those perturbations and so on. Like it's a very important practical task. But the key challenges to develop those models is to actually define what processes might underlie those properties of microbial communities, and how to describe them as a set of rules or ecological laws, and then like where to get all those parameters to put in those laws. And to start with, the simplest way to describe microbial communities is to just take a species into account and then define some interactions between them. And this is like another very popular model that we're not really touching in this consumer resource stuff, but like I would say it's like 50-50 between Lotka-Volterra models and consumer resource models in my subfield at least. And in Lotka-Volterra, what you actually do, you have this a term that basically resembles how do different species affect each other growth. And it either can be a positive effect or a negative effect, and like negative called competition, positive called cooperation. And you actually can infer all those things from the data you collect in the lab, and then hopefully you can predict community dynamics. And those models were proven to be working to some extent, but they actually have a lot of drawbacks, like you have no information what actually underlies those interactions mechanistically. And this model only gives you certain hints that those two microbes have a very strong positive link. There should be some cooperation. Maybe I should do some additional experiments and dig into what's going on. But to some extent, those interactions are not very reliable, and people recently were showing that sometimes you infer those interactions from the data and they just don't work. So community shifted to the next kind of level of detail to describe those communities. And they realized that like a lot of microbial interactions are actually mediated by metabolites that are there in the environment where microbes are residing. And in fact, those competitive interactions typically arise from competition for resources. And these cooperative interactions, they can be mediated via cross-feeding, when one microbe produces some metabolite and another microbe consumes it, and then like they both boost their growth. However, for those models, we actually need to collect a lot of data, because if you really want a model to properly describe your experiment, you need to know how exactly those microbes grow on each resource. And you also need to have all those growth parameters. So it actually involves not only measuring microbial growth curves and like microbial abundances in the data, but you also want to have some idea about what metabolites are there in the environment, what are their quantities, and so on. And actually, models that describe this species-resource interactions, they existed in microecology for quite some time. They were developed in the 80s for like some plant biology or like predator-prey ecosystems. But then they were mostly staying out there in this theoretical field until people from microbial ecology actually realized that, look, a lot of microbes actually probably follow the same ecological laws, but in microbial ecosystem, we actually can go and directly test it in the lab, and it's going to be fast and easy comparing to this large macro ecosystem when it takes years to collect the data. So like in the past like 5 years, there was like a huge surge of those consumer resource models in the literature, and people started to study various scenarios on how they work in microbial case. And most popular scenarios, is there any question? Okay, that's just some noise. So most popular scenarios that people study with those models is the following. One is the community assembly, when you have some global species pool, and then you have like invasions of species to some local environment. And succession is a part of it, because there is a notion that some microbes can actually come in early because they can digest primary metabolites that are there in the environment, but then they will just produce some byproducts and run out of their primary resource and just probably die out. But those byproducts might be a new niche to occupy for some other microbes. So like we can have like all those waves of blooms and extinctions in the community. Another very interesting and practically important scenario is community coalescence. It's when we actually mix two different communities and see what microbes from each community will survive. And it actually studies like whether we have some, it's called like metabolic cliques in the microbes, meaning that like sometimes they just really form those tight cross-feeding communities that support each other, and then they can invade in the environment as a whole. They can't really survive without each other in any given community. And of course, like a large portion of this research goes into studying what are the optimal strategies for microbes to grow under different conditions, what parameters can affect your model, and so on. And the core assumption for all consumer resource models is obviously how exactly your microbes will utilize resources. And as of now, there are like 3 major groups of assumptions. One is co-utilization of substitutable resources, and this is like a most popular one, so we're going to start with that. Another one is deoxy of substitutable resources, which means that microbe will switch from one resource to another and consume just one resource at a time, one by one. And the last one is co-utilization of essential resources, which means that microbes will actually need both resources in order to grow. So with that, let's move to MacArthur model. And the formal equations that describe this dynamics look like this. We have 2 major parameters here. One is this lambda, another is this yield. Lambda actually describes resource affinity. It means like how good is your microbe at uptaking this particular resource. And yield quantifies the amount of biomass that you can generate per unit of this resource. And this delta here is like some removal of microbes from your system. It actually can be mortality rate. It can be just dilution if you're operating in a chemostat or in a batch culture or something. And in a lot of cases, especially in the chemostat, like delta for microbes and delta for resources, they're the same, and it actually allows you to save a little bit on parameters in your model. So on the resource part, you actually have influx of resources because resources have to come from somewhere. Sometimes here people put some resources that actually self-reproducing, and it was inherited from macroecology, but I think in microbial communities, it actually just makes sense that you just have some supply of resources like in a chemostat, you just have inflow rate. And then like you have this term for consumption where you have like sum of all resource uptakes by all microbes times this resource concentration, right? And like on the left here for your reference, this is how your growth profile would look like. You will have some nice saturating curve and like all resources will be consumed simultaneously. So they will just decrease together with like different slopes here because like you can have like different coefficients for their consumption.
So Veronika, can I ask a question? If you have an essential resource, it doesn't really matter, right? Because it like you would still be able to grow if it's absent because you're just summing over other resources you use. Is that correct?
No, I think in this model, we are actually ignoring those essentiality for now. Like under MacArthur assumptions, all resources are substitutable. It means that each microbe can grow on each one of those resources. Like
Yeah,
if all others are absent, it will still grow because this is the sum, right?
Yeah.
And typically, this model is used for some experimental settings where you have like large amount of other resources and you're just checking like different carbon sources or something like on a rich media with all other resources supplied, right?
And
so,
the dilution rate is independent of the rate of the growth rate and stuff like that. It's just like a okay.
It's independent, but it actually comes in here because in order to find your final steady state, you will have to balance your growth with this dilution, right?
Oh, yeah.
Because like if you grow slower than dilution, you will eventually be washed out of the system, like go extinct and die, right? So like this term will like in the steady state, it should be equal to delta. And typically like what people do from this microbial biomass equations, they get the steady state concentration of resources. And from this resource equations, you just use those steady states, you put them in here, and you get your microbial abundances or like bacterial population in whatever units you defined. And the core thing to know about those communities that are described by consumer resource models is that just by definition and how we import all those parameters in the model, we will have this universal limitation that the number of surviving species in the steady state cannot exceed the number of resources that you have in your model. And why it is like that? Because in a nutshell, if you consider this very simple case when you have two resources competing for one resource, two microbes competing for one resource, whatever microbe will have higher growth will eventually win because it will accumulate its biomass faster and like consume resource even faster. So another species will just die out throughout your dynamics. And for quite some time, people were actually a little bit upset about it in this microbial ecosystems field, because we definitely know that for microbes, it shouldn't be true. Because in a lot of environments, you actually observe a lot more species and resources. And there are experiments when you can have like multiple species surviving on a single carbon source or something like that. So people started to develop all those extensions of classical MacArthur model that deal exactly with this problem. One is that you start to introduce byproducts in your model. And byproducts is like a first solution if you have like one primary resource and you basically artificially extend your resource space in order to accommodate more microbes in your system. And it actually adds two more parameters in your model. One is the leakage fraction. It shows you how much of your primary resource goes to your biomass and how much is excreted as byproducts. And another one is like this stoichiometry of conversion between your primary resource to a certain byproduct. And the only thing to take into account here is that you have to make sure that when you specify those parameters, your amount of byproducts should not exceed your amount of like and energy yield of your original primary resource. And this is like a most popular variation of MacArthur that is widely used in papers that are published recently. And I'm going to give you like some examples how it actually works and what are the consequences of the modeling your community like that. So that was actually a paper from our lab from like a couple of years ago when we tried to see how this trophic chain of microbes establishes when you have this assumption that each microbe has one primary resource it can consume, and then it just generates like a couple of byproducts. And what we simulated here is that this is your time scale where you have like invasions here. And in some invasion events are actually successful because like if you get a microbe that can use the primary resource, you're good, it's gonna stick there in the ecosystem. Then whatever microbes that will be able to utilize the byproducts that's synthesized by this first microbe will also stick in, and so on. You have like this whole chain. And the interesting part of this dynamics is that actually sometimes this microbes that came second or third or whatever, it might be replaced by some other microbe which has a higher growth rate on this byproduct. And then you will have like the whole avalanche of extinction because all those microbes will also go away with this first branching point. And the longer you run your dynamics, the larger will be the sizes of those avalanches. But they will be like more and more rare because like towards the end, you will probably go through most of your species here. And another nice thing that you can understand from such a simple thing is that microbes that goes on top will have a higher abundance in general. And in the end, if you collect all of those time points throughout your dynamics, it's most of those microbes that come on top, they will be most prevalent because they will be present in majority of points here. So this is actually resembling a lot of trophical chains that you have like in natural communities, because like in the human gut, it's known that you have like primary degraders and they will be having larger abundances than other microbes. And also like it's known that in a lot of cases you have like primary degrader and then it will have its whole group of species that it actually can support. And those species will not be there if you don't have this primary species. Another nice paper that studied this model was actually published in Science in the same year. And that paper was super cool because they actually tested experimentally that on a single carbon source, you will have a pretty high diversity of species. And they confirmed that those species actually grow on byproducts of like couple of primary species. And they actually used MacArthur model with byproducts to model qualitatively what's going on in their experiment. So I really recommend you to check out this paper. Another important thing that people typically discuss in MacArthur is that those parameters should not be independent. And why it is like that? Well, first of all, this uptake flux has a actually practical limitation that comes from the following thing. Typically, uptake is defined by your transporters on your membrane, and the surface of the cell is limited. You can't really have all possible transporters in high quantities on your membrane simultaneously. You don't have the capacity. So each microbe will actually have to choose which transporters to keep in order to maximize this growth function. And the same goes to those yields, because in those yields actually is inherently included this cost to synthesize all of the enzymes to digest those metabolites. And you also want to optimize for this cost. So in a lot of models, people actually add this additional assumption on enzyme budget, which goes as follows, that like the sum of those parameters should be equal to some constant for all microbes with like slight deviation. Basically this assumption gives rise to the following interesting scenario, because when each microbe decides how it will place its beds, it can pick between two different strategies. One is you put everything in one resource. It's called specialist strategy. And then you will actually probably maximize your growth as much as possible, because you just pick whatever resource you have like highest uptake and highest yield, and you're good. And then there is a generalist strategy, which means that you will consume a lot of resources simultaneously, but you won't be really good at any of them, and like they will have like marginal effect on your growth. And from a certain perspective, if you have like very stable environment, typically those specialist species win, because they will actually maximize their growth as much as possible. But then if your environment is changing and it's like a setup called fluctuating environments, the generalists actually have a lot of advantage, right? Because the specialist species won't be able to survive in the environment when they have like large quantities of those other resources that it actually can't utilize. But generalists will be just fine because it can grow pretty much on everything reasonably well. Any questions so far?
Sorry, I'm asking all the questions. So in the dioxy model, you can't have loops, right? Because you just have one resource. Is that correct? So in the like
in byproducts model?
No, in the dioxy.
Wait, we're not yet there in dioxy.
Just one before. Oh, you're not in the interaction yet. Okay.
Yeah, right now we're considering that like each microbe can consume some arbitrary number of resources, but it consumes all of them simultaneously. And you can see it in experiments that like you can mark different resources by some technique and just literally see that it consumes this and this and this, like their quantities are running out at the same time.
Yeah,
This is the MacArthur.
I was just wondering, like, if you could in those branches that you showed in the like 2 slides ago, if you could get any kind of loop there, because
in principle, loops are possible. Yeah, I don't know where you're going from. Like in human gut, for example, all this cross-feeding actually looks like this sometimes, right?
yes,
Like,
and also there's these like model ecosystems that they keep perpetually, because you have someone that's a primary producer and then a secondary producer, and then they can keep it for years, for example.
yeah. MacArthur with byproducts takes care of that situation, because basically as soon as you establish this first species, it will start producing those byproducts, and then like you have those microbes and they will synthesize something else, and then it can contribute to these terms again. And this whole resource dynamics actually can be really interesting, and I think we are going to go through it in the practical part of this tutorial, because if you actually will track dynamics, you will have something like this. Your primary resource will go like this, but then your byproducts start accumulating, but then they will start to be consumed and something like this.
Yeah, and I would expect that to be happening in that Go Forward paper that you showed, right? That you have these, that they just keep the community going without putting a additional carbon source.
Yep.
I'm a bit confused. So this is without the enzyme budget trade-off or the membrane trade-off, right? So why doesn't like the first species also eat its own byproduct? Why would a second species go and do that?
Because under MacArthur assumptions, what you actually have is the following situation. This species, whatever it secretes, it doesn't need. So we're ignoring what's going on inside the cell.
Okay, so that's an assumption I missed then. Okay, thank you.
Yeah, I understand that like in those flux balance, what you care about what's going on inside, and then like whatever you synthesize, it actually you use it, right?
But I would like to comment on this, because I had the same question regardless of flux balance analysis, because some species do take up what they produce, right? So this happens a lot. So it is maybe a fundamental question at which point the species recycles before another species can use up what happens, and how that is decided, whether the species recycles or whether another species comes in and consumes.
Yeah, I think my understanding is the following, that all those byproducts, they actually can be used by the primary species, but they stay inside, right? And we're ignoring everything that's inside. So whatever leaks here as byproducts, it is excess. So this primary degrader has no way to effectively utilize those resources. That's why it just outputs them as a waste for himself, but other microbes can benefit from it.
Yes, but we observe this to happen, something that has been secreted being consumed again. This can happen, right? So this is something.
Yeah, I think that this is a very complicated scenario, and this is an extension. And in the classical MacArthur scenario, it actually is never happens, I think.
Okay,
yeah,
so we hit on the limitation already with deoxy shift in E. coli when it, I think it uses acetate and then starts consuming it. And
Yep. I know where you go coming from this fermentation versus respiration and all this stuff. But no, MacArthur actually is not about that. It's very simplistic thing.
Yet another limitation that maybe can discuss here is about internally that species do different things in the presence of other species, right? This is maybe also something that we assume that they are constant and don't change strategies, which actually they do.
Yep. MacArthur can't really take care of it. And I think this is also a major flaw that happens in this lot covalter, right? Because depending on other community members, you can have different interactions. So yeah, MacArthur has the same problem, that you can't really account for those changes when you have different community members and you're changing your strategy. Yeah, typically we assume that there is like one general strategy for the resource, that if it has this primary thing, it will output that byproduct. And this is it, but it's set in stone and we're not really changing it.
One last question on the length of the chain. So when you show the branches in that paper, paper, I think, so then how do you know how many branching points you can have? Because in GATT, we do not have all that many levels. And I'm not sure you can call them trophic levels. That's really if one organism eats another one. I don't know anyways, but how many of those levels can you have? It
actually depends on dilution. And I think in the GATT, it's also like that, because if you do it with pen and paper, it will be like very obvious to you. But in general, because there is this whole balance between how fast you grow and how fast you get diluted or die. Like as soon as you have a resource concentration that can support enough growth, you're good. The species can stick in the community. But if you start to increase dilution, you will start to get rid of all those microbes that come later, because at some point there will be not enough resource to overcome this death and removal from the population. And actually in the gut structure, I think it might be mediated by this gut motility things that people started to study at some point, right? That different people can have different motilities, so you can actually have different number of layers in this chain, just because this motility actually sets your dilution to some extent. But yeah, that was a really good question about those all things. So yeah.
Can I ask a question?
Yep.
So in this MacArthur model, can you also model succession? So for example, if your primary degrader stops becoming the primary degrader because you have a perturbation or something.
Yep. Yep. It is what it's used for in a lot of cases, right? Okay. Yes, you can have this community assembly, and in this community assembly, you will have this natural succession, because you will see that this microbe sticked in, it produced its byproducts. Now, this primary resource can run out if you set it up like that, that you don't have like this constant supply of resource, and so on. And then you can have like all other microbes coming in on byproducts. And yeah, you can have all funny dynamics in there.
Okay, thank you.
Okay, with that, let's move to the second type of the resource utilization strategy, which is sequential utilization. And this sequential utilization, it's typically called dioxy or multi or polyoxy if you have many resources. And it was actually discovered way back in the 50s by Monod when he studied growth of E. coli on different sugars, because he noticed that actually what happens is that E. coli seem to grow on glucose first, then you have the slowdown, and then there is the second growth phase, and like there can be another slowdown and so on. And it actually, if you will measure how your resource concentration looks like, you will realize that during the first phase, you actually deplete just one resource. Then during this lag, no resource is depleted and cell is probably changing its strategy. Then second resource starts getting depleted and so on. And this strategy is actually very widespread in the nature. And recently there were some papers where people started to measure experimentally those dioxic phenomena for soil bacteria, for gut bacteria, especially on these polysaccharides. Also there were some papers where they detected that there is some deoxy on amino acids. And the main benefit for the microbe in this strategy is that it actually resolves this enzyme budget limitation. Because you, as a microbe, you can actually invest everything in your most favorite resource that gives you highest growth. You can flourish, then you can have this regulatory switch to the next preferred resource, and so on. And for quite some time, people were mostly working just on models, like how to properly model these growth curves and like how to take into account these regulatory events. Basically, experimentally, the most interesting thing to study here is that what are the preferences of different microbes? Because it seems that some preferences can be universal, like this preference for glucose, like every microbe will go for glucose. This is a very energy-rich source. This is a no-brainer. You want to prefer it. But then for other resources, there might be actually discrepancies and different microbes prefer different things. And moreover, as Daniel mentioned, you can actually change your preferences depending on other members of the communities. So this is like a very rapidly developing field of study. And for deoxy behavior, the core thing to take note of is that there is actually a trade-off between your growth on your preferred resource versus the lag after that. And why it is like that? First of all, this is like one of the recent papers where people again confirmed it. They had like different Saccharomyces strains. They demonstrated that those strains actually also have deoxy on a mixture of sugars, which is glucose and galactose. And moreover, they show that different strains can actually have different growth curves with more or less pronounced lag phase here. And like from those growth curves, like the growth rate is actually this slope. So they measured all those growth curves, they quantified growth and lag, and try to dig into detail why there is this big variation here. And it turns out that if you look at the protein that is actually involved in the galactose utilization, you will see the difference between those two behaviors. And here they show like two most extreme cases. One is when microbe is actually utilizing glucose until the end. So here you see that this is a time point when your glucose concentration runs out. And after that, you still have this time when you don't start to produce this protein, which means that your switch started to happen just somewhere here. And then like you have this transcription, translation, finally you have this protein, and now you can start utilizing the second resource. And so it starts to go down somewhere here probably. But another strain was a little bit smarter. It actually started to prepare for the switch earlier on. So like you see that here at this point when the glucose just started to run out, but not there yet, it already started to produce this galactose protein. And if you look at the growth coefficient for those microbes, what actually happens is the following. This microbe that prepares early sacrifices a part of its growth because it starts to repurposing a part of its enzyme budget to prepare for the next resource. But then when this resource runs out, especially in the co-culture scenario, it will get a big boost of its growth. So eventually it will have a higher population, if you will, co-culture those two strains with different strategies. And basically, in many other papers, people were showing that this growth lag negative dependence actually happens. For example, for E. coli, people were measuring like this growth on the primary resource versus lag when you shift to another substrate. And there is this nice dependence, no matter what is your primary resource. And like when you switch to acetate, they will all be pretty much on the same line. And it's always negative. On different resources, the slope of this trade-off can be a little bit different. It might depend on those enzyme budget costs and so on, but nevertheless, it stays there. So when we parameterize this dioxic models, we have to take a note of that, because like you can't really generate those two parameters independently. And we also contributed to those dioxic models because for quite some time we were wondering what will happen if you have multiple microbes which all can shift dioxically between their resources. For some reasons, there were no models like that in the literature. Everyone was just studying this growth curve for one species. So we developed our own model for this scenario. And it actually has like two main parameters. One is you have to specify this preference order of resources for each microbe, and we allow them to be different. And another is that you can have different growth rates on different resources, and sometimes they can be actually correlated with those preferences, but sometimes they can be varying, and we also know that it happens in nature. So we actually tried to do this community assembly experiment in the popular batch culture scenario, where we have this batch culture with different carbon sources, and we have microbes coming in one by one, and we cycle the model until each microbe has a chance to be established or to be washed out. And basically, key findings of this model is the following. The whole selection for community members will have on the top choice resource. And if you think about it, it seems logical, because the top choice is what kind of sets your initial population. And like as soon as you grow really fast at first, you will reach more much higher abundance, and then like even if you grow not as fast on your second one, you will still have this initial boost. And like in this whole dynamics process, you will still get a bigger chunk of resource and so on. Another thing that we noticed is that when invaders come in our system, they actually typically come on a resource that is currently utilized the least efficiently. And this is also kind of a very natural thing. But what it actually leads to is the following, is that in the final communities that gets established, typically each microbe will have a different top resource. And we will have this nice separation of resource niches. And we think that this is a very cute result for such simplistic assumptions. Another important thing to note here is that actually there is a possibility to have a mixed strategy in all those consumer resource settings. And mixed strategies between this switching behavior and co-utilization. And there was this paper from Chao Tang's group 2 years ago, where they actually showed that this choice between dioxy versus co-utilization of resources actually comes from the topology of your internal metabolic network. And I think that every crowd should really appreciate it, because they actually argued that, look, you have here like this whole glycolysis pathway, and basically all those sugars, they come into this glycolysis here. And because they all go through the same chain of reactions, for a microbe, it actually makes sense to utilize whatever resource gives the highest growth and try to ignore all others here. So you want to maximize your glucose uptake as much as possible because it will give your highest growth rate. So you want to downregulate all those other enzymes that can potentially block this flux here. But then you can have another set of substrates which actually come in here in those cycles which are downstream. And those cycles are kind of core, contain core metabolites that cell will be dealing with anyways, right? So because they're all synthesized from this glycolysis, and if you actually also provide them in the media, it's possible to boost this whole metabolic cycle even more. So they did some experiments, and they actually confirmed that indeed, like, microbes will switch between those sugars, but if you supply sugar with some downstream metabolite, they will be actually co-utilized. So I think this is a very important thing to take into account, that dioxy and co-utilization are actually good strategies for different kinds of resources. And like when you try to model your microbial ecosystem, you should think which metabolites are there so that you can properly incorporate the strategies in the model. Are there any questions?
I would say there's maybe one situation in which this is not optimal. If you are producing something that another microbe takes up that also competes with you for another resource. In that case, it's better to just as quickly as possible consume everything, all the sugars.
Yeah, and this is what I tried to hint at when I was describing that, yeah, microbes are changing their resource preferences depending on other members of the community. At least there were several papers that people reported it, but I think for now we don't really have a general understanding how universal it is, and like how to get any idea about it without really going and measuring it, right? You can't really know what are the resource preferences of a microbe just from this genome annotation or something. You have to go and make sure, and you have to do it in a co-culture of microbes and so on. Because, yeah, people were doing those experiments and they actually checked that if you grow a microbe as an isolate, it will have one sequence of switches between resources. If you start to co-culture it with another, suddenly the order might slightly be changed. And yeah, I think this is a very under development set of experimental studies.
Yeah, I agree completely. Thanks.
In this example that you gave, would you expect these additional resources to be like boosters, or like would the growth rate be the sum of using these resources, or would they just be an additional boost if they're using their preferred resource?
In principle, my understanding is that at least authors of this paper considered it as a, like, if you have resource from group A and resource from group B, this is a MacArthur scenario.
Yes.
Basically. So like you will have some growth just like on pyruvate, you will have some growth just on glucose. And then like if you have both of them in the media, you will have a sum.
Just a linear combination.
Mm-hmm.
Yeah, but then if in the case of a boosting thing, you would have something like I don't know, like if it only works if that is present, then you have to kind of multiply it or something.
Mm-hmm. Yeah, this is a possible of growth function that you can specify for the model. But I think that at least for those metabolites that they specified here, those are really core things. I think it as long as your bacteria grow in your experiment, it will synthesize all of those, right?
Yes.
No matter what original substrate you had. So they will always serve as boosters if you try to add them to your environment.
Yeah, the only question is if it would be like the sum of the, like how much you use of one and how much you use of the other, would that sum be your growth rate? And if they are individually or not, like it's only a booster in the presence of the other resource.
Yeah,
that's what I think it's
so.
complicated in making the model.
I think so. But my impression, at least for some papers, people actually sometimes use pyruvate or succinate or malate as the only carbon resource, and cells still grow. So I think because they still have some energy to be extracted, right? It's not CO2. You still can oxidize them and get something out of it. So it's possible to grow on them. It's just you will grow slower. Okay, so we're hopefully done with deoxy and we can move to essential resources. So this whole notion of essential resources is actually coming to the microbial field from the plant biology. And in the 80s, Tillman actually wrote the whole book describing how plants, and then he generalized to microbes and other species, actually require not just one resource to grow, but multiple others. For example, like carbon, nitrogen, phosphorus, some microelements. So this growth on such a set of resources, it's not just a linear sum. It should be some multiplication or something else, like some other functional forms that actually accounts for the fact that you can't grow without one of those resources. And typically the model is formulated like this, and it looks very similar to MacArthur. It's just like here is an abstract form for some growth function that should depend on all of the essential resources. And then you also have this consumption term for all of these resources. And the simplest function that people typically use in this literature is the mean function. And this mean function has this intuitive explanation, is that if you require a certain amount of, say, nitrogen and certain amount of carbon in order to grow, then if you dump like more carbon to your system, it will not really help you to grow more, because you will be limited by that other resource, and nothing can actually be done about it. Typically, the resource that is the least abundant in this min function, and that will be picked as a result here as the thing that actually sets your growth rate, is called the limiting resource. So typically, if you measure how your bacteria consumes resources, unless you will match your resource concentrations such that they match exactly your required nutrient stoichiometry, you will probably just consume all of the limiting resource, and then you will have some leftover of some essential resource. Basically, there are some other growth forms that you can encounter in those papers. One is this multiplicative one. Another is this monod style with saturation. Another core thing that people use to describe those ecosystems is that resource plane. And it's actually a very powerful thing to develop some intuition about those resource limiting environments, because it actually shows you how your ecosystem dynamics will look like. So basically, what is shown here is that you have, for a set of two resources, as this is a most simple scenario from Tillman, you will have this 2D plane, and your additional resource supply can be somewhere here or here, depends on your actual environment. But then if you have just one species, it will set some minimal concentration of resources such that this is the most extreme value until this resource will be depleted at a steady state. And this angle is for the mean function, but for other functional forms, it can be something like this or some other functional things. So basically, if you populate your ecosystem with species B, say, it will start depleting resource, something like that, according to your parameters, and then it will end up on this line somewhere at a steady state. And at this steady state, if you remember, it is a situation when you have your growth rate to be equal to the death rate. So it's called the zero net growth isocline. So on this line here, the microbe doesn't really grow anymore. It just barely maintains its population, and it can never actually cross to this region, because here it will just die out. So if you have more than one species in this scenario, you just add this other angle here, and basically the intersection between those two angles will be your community stable point. And no matter where you start, you will actually arrive here, because all those people like to draw all those phase diagrams that look like this, that you will go here and here. Yeah, but basically you're guaranteed to end up here. In this scenario with two essential resources, people actually discovered the following trade-off, that in principle, you can imagine that different microbes can have slightly different stoichiometries in various essential nutrients, because they can somehow optimize their composition of atoms in the cell by optimizing some protein amino acids and stuff like that. And then you can have this whole zoo of angles here. This is the most extreme point where each of those microbes can survive. So like if you start to throw in more and more microbes, they will end up on a line like this. And like many microbes will be here on the Tillman plane. And those intersection points will be your possible community states that will be realized in different environments. But if you will have this fluctuations of resources, you actually can have a transient state where many of those microbes will be there in your ecosystem. It's just some microbes will be on the way to die out, some microbes will be growing. But if you just take samples from your environment in different time points here, you will detect all of them. So I think this is like a very nice setup for many natural ecosystems, because we know that you can sample multiple points in your soil field and then end up in slightly different compositions of bacterial abundances, but nevertheless, the pool of microbes will be similar. The same goes to human gut and so on. So it seems that microbes actually can be living in those fluctuating environments under all those nutrient limitation constraints. And majority of the papers about Tillman model, like honestly, are very complicated and they're very theoretical. So I don't really want you to dig into details of that during my talk. But one small thing to highlight is that if you were wondering how MacArthur assumptions will look like in this resource plane, they look like this. They look like lines. And basically, those points are also defined by this steady state resource concentration. So here will be R star 1. And this R star 1, you will reach in the absence of the second resource. So in a nutshell, it's going to be just your dilution or death rate divided by your growth, which is lambda and yield. So, and then like if you have several microbes, this intersection is actually what sets your community. And moreover, like this is where your community will set steady state concentration of resources. And if you want to have more than 2 microbes in this community, you will have to make sure that all of them will, it's hard to draw here, but they all have to go through this point here. So as soon as they go through this point, all those microbes can be here coexisting in this community. And basically, if you contemplate on it really hard a little bit more, you will understand that this assumption about all microbes going through this point actually arises from this enzyme budget constraints, that like all of the lines that go through this point, they have the same enzyme budget. And for basically for Tillman's plane, there are many extensions, and for Tillman's model, like some extensions include more than 2 essential resources, and then this whole thing gets really complicated because you can have like all sorts of cycles, chaos, and so on. We built our own model where we introduced multiple resources of each essential type. And then we ended up with a very complicated model where we have multi-stability, hysteretic transitions between states, and so on. So it's definitely out of the scope of this talk. But this is basically one of the last things I wanted to show you from the experimental part. Basically, this whole resource limitation is mostly studied in marine ecosystems, because in marine ecosystems, people confirmed that they are severely limited by many resources like nitrogen, phosphorus, iron, light sometimes. So in like various parts of the oceans, there can be like different limitation conditions, and then you will actually observe different communities. And if you bring a set of like typical phytoplankton species that reside in the ocean and realize different resource limitation in your lab, you will actually indeed see that in different resource limitation, different species will take over. So it really hints that typically like those species are limited by a certain resource, and when you take samples and do some like metagenomic analysis, whatever abundances you get may vary a lot due to the environmental limitations. So what I really want you to take home from this theoretical part is that we can have like variety of growth strategies in microbial communities. In various scenarios, different strategies might be worse or better because there are some intrinsic trade-offs, and environments also may vary. So microbes really have to adapt and use different strategies. And consumer resource models are actually simple, but yet they allow you to explore multiple of such scenarios. And there are many assumptions that are not included in consumer resource models yet, but people kind of think about them. They try to discuss it and sometimes try to incorporate them to some extent. For example, some of the byproducts that are generated by microbes can be toxic. Like some microbes can synthesize some antibiotics, or some of the byproducts can actually change pH in the media, and then it will actually affect our other members of the community. You can also try to put phages on top of this community dynamics. And the simplest way to do it is to include them in this dilution factor as like additional mortality term. But that's true only for lytic phages, right? And for phages that also have like variability in strategies like lytic and lysogenic, it's not yet so simple to understand how to actually put them in here. Then this whole regulation of growth strategies is also under development. People have proposed several models that try to incorporate them, but typically they increase complexity of your model by a lot because you have to put in some additional parameters on this regulation. Another interesting assumptions that you can add in the model, and I think it partially relates to this switch between fermentation and oxidation and so on. So sometimes microbe can actually store resources for the future. Like you can synthesize something and then you can store it inside the cell, or you can put in the environment in order to consume it later on when you will have the opportunity. And another thing that is totally ignored by consumer resource models for now is spatial structure. I think it's a very intricate balance between going from this very, very simple models with as few parameters as possible towards this whole zoo of like agent-based stuff, flux balance. So like you really have to put a lot of thought into what you want to capture and what should be important for that. Like for many ecosystems, the space might not be that important, like for marine ecosystems or for something else which lives in this well-mixed diluted setup. But then, like, many microbes can live in biofilms, many microbes can live in something like soil or human gut, where it actually makes a lot of sense to collocate microbes that do this cross-feeding and so on. So it might actually result in additional parameters in this model. And I think with that, I want to end the theoretical part, and hopefully I did not confuse you completely with all those things. And please ask me any questions if you have. And yeah, I actually wanted to quickly advertise one other thing. On Fridays, there will be a set of talks by Alvaro Sanchez, Guy Bunin, and like other people who actually do a lot of model development in this consumer resource field, and they do a lot of experiments to validate consumer resource assumptions. So if you're really into it, you can go and check it out. It should be a very nice set of talks to listen to.
All right, thanks a lot, Veronika. A great talk. Is it open for people to register in this?
Yep, it's open. You just go there and they will send you a Zoom link.
So we have like the final session for questions, unless someone has a burning question to ask right now. Is there anyone want to make like a question before we move on? If not, then we can take a break. The idea is to take a 10-minute break and be back at 4:30. Yeah, where we go to the practicals. Is that okay? All right, then see you guys in a bit.
See you.
Part is a total experiment on my part. For consumer resource models, I would say it's particularly hard because every person who develops its own model, it just writes its own code in whatever language he prefers. So in every paper, there is like attached GitHub, typically with like code on MATLAB, on Python, on Mathematica, on C, and so on. So the only thing I managed to find is that like last year, guys from Boston University actually compiled some more general Python code that allows you to play around with several consumer resource models that I described in the theoretical part today. So I just think that it might be a useful exercise to just walk through their functionality of their package and see what kind of parameters you need to specify in order to run community dynamics, and what kind of scenarios you can explore, what kind of plots people typically have. And hopefully it might be useful to some people who do some experiments and they just want to jump onto it and do some simple modeling that will recapitulate some of their experimental findings or something like that. So basically, hopefully you all can just get access to this Jupyter notebook. I rarely use it in my work, so I'm not that familiar with it. But basically, for their model, they define several sets of data classes and functions that can use those data classes in order to do the modeling part. So they have classes that are called plates, And they designed it for this typical experimental setup when you have like 96 well plate, and like in each cell here, you put some media with some bacteria, and then you just propagate it in whatever setting you are studying. In this plate, you basically have to specify your resource amounts, like initial, at least your initial bacterial population, like what bacteria you're putting there, and like your initial quantity. Then we will have to specify for this plate class dynamics law that will describe growth rates of your microbes and like how do they consume resources and how they run out. This all can be summarized in this parameter vector that is specified for this plate. And then they developed this function called community. This function community basically grabs your initial state for the plate, your dynamics laws that you specified in the functional form, your parameters, and it just runs your simulations. So for the first thing, let's basically, okay, fine. This is their example. For now, let's ignore that they have their some default assumptions that they put for MacArthur consumer resource model. Basically, what they had to specify is like how many wells you have and what is the leakage to byproducts. Then you can actually specify for each well what is your resource gradient. Yes, like you probably want to run like 100 different environments to test. Basically you input this vector of initial resource supplies into your assumptions. This make initial state function grabs those assumptions, makes this data structure that will specify your microbes, specify your resources, taking this parameter for growth into account. Then they have this function called consumer dynamics, which is, in this case, it's MacArthur with byproducts. And here we just run it, and this find steady state outputs your final microbial and resource concentration for each well in the end of your dynamics. And basically you can plot this typical plot of stacked bacterial abundances in each well, and see that the more resources you have, the more bacteria here you will eventually establish in your community. And another thing to take note that in all those dynamical simulations, typically you will get many bacteria with very, very low abundances. And in a nutshell, you probably want to put a threshold here because those bacteria are not really surviving in your ecosystems. This is just some numerical thing which arises from your numerical integrations. But basically, if you think that this is like log species abundance, bacteria which has abundance 10 to the power minus 6 is probably undetectable in the experiment. So like in experimental data, we never see those bacteria. So you want to really work with this tail of this abundance distribution that contains bacteria that has like noticeable abundances. Okay, so basically this plot tells you the summary of this picture, because here it's sometimes really hard to see how many species exactly you have here in each well, but this plot shows you that like on a very low resource concentration, you will have like 1 or 2, 3 species surviving, but then if you dump a lot of resource, you will allow for establishment of this long trophic chain with lots of byproducts, so you can really boost your diversity in your ecosystem.
Veronika, are these types of resources or concentration of resources? I'm sorry, I missed this.
Here is concentrations.
Concentrations.
Yeah, a food supply level means concentration of resources, like what amount of resource you put in each well. And yeah, for resource supply, let's dig into detail. Basically, this is how all possible set of parameters that specifies this MacArthur model looks like. First of all, you have to specify the size of your species pool, like how many species you want to have in this global pool. Another is the number of resources you want to have in your model. In principle, you can have more species than resources because like typically you generate them randomly. So some species are not really competitive. So they will actually never stick in your ecosystem, right? And also in this scenario, you actually can study all these community assemblies and successions when you can actually see that microbial fitness improves, that like more and more competitive microbes come in and they cause some extinctions and displacement. So like typically you want your species pool to be higher than your resource pool. And also for sampling of species from your species pool, you also can use like various popular methods to do it. Like most simple is binary, but it can be a Gaussian or something else. One confusing part for you here is probably what is this specialist family when they define their species. Here in this example, basically what they do, they assume that your resources can be of different classes, say like sugars, then proteins, and maybe some lipids. And basically you can have species that are specialized on a certain class, and then they can produce like another class as byproducts or something like that. So basically this class specifies like modularity of your consumption metrics, because in the nutshell what you actually do, how you define your species, for each species you define this table, right? This is your species, this is your resources, and in each cell here for species, you say whether the species can consume this resource and what are the like lambda and yields for this specific resource, right? And basically here they want to work under the assumption that you have like some clusters in your consumption matrix like this. Another parameter that you have to put in here is the distribution that you are going to use to generate lambdas and yields. Ideal situation, of course, when you can directly measure the lambda and yield for each of your species in the experiment. But in a lot of cases, people are actually limited in their experimental capabilities. So they just generate a big pool of species, then they run those simulations and select like whatever simulation resembles what they have in the experiments. And then they say that our species probably have parameters similar to this. And the smartest assumption you can actually make is to have some idea what is the range of those parameters which is feasible in natural ecosystems, right? But that's pretty much it. Another parameter that can be specified is whether you want to focus on specialists or generalists in your model. Very, very important parameter is the leakage fraction, because the more you increase this parameter, the higher is your diversity. And if you put leakage to 0, you will have like very few species surviving, and each of the species will be like the highest growing resource, highest growing species on each resource. Those two parameters are very specific to this specific form of their consumption matrix, because they are showing like how the conversion between different types of resources happen, right? This FS shows you like that you can actually produce byproducts that belongs to the same class. And another shows that like you can produce byproducts that actually belongs to a different class. A sparsity is also specifying this matrix, which was stoichiometry matrix di alpha, alpha beta. This matrix shows you like how do you convert one resource to another. So because you can imagine that you can't really do a conversion of one metabolite to all other metabolites, right? You probably can have like several byproducts that arise from one metabolite, several byproducts that arise from another metabolite. So this sparsity actually tries to take into account that not everything can be converted into everything. So in order to maintain realistic scenario, you probably want to maintain the sparsity of this metabolic matrix to be low. This parameter is basically specifying like how many independent simulations you want to run in one batch. S specifies how many species you want to put in a given well simultaneously.
Veronika, may I ask a question about the sparsity matrix? Can be put in prior knowledge also? Do you know?
In principle, it's possible. Yeah, if you have an exact knowledge of what is your metabolic matrix and which metabolites are result in which byproducts, yeah, you can directly specify this matrix. Here, they provide just like a dumb example where you don't really know how this matrix look like, and you just want to generate some random matrix with just sparsity similar to the real matrix, but that's it, yeah.
Yeah, and one more question. I think I missed what the sampling algorithm was all about.
About what?
The sampling algorithm, so the very first one. I think I didn't really get why you had, what it was doing, what was the idea on.
So the sampling basically is the following thing, like your species pool can be really large, right? And sometimes you might want to put subsets of species instead of putting everything from your species pool. Because if you will put all microbes from your species pool simultaneously, you will probably arrive at one steady state, which will be some like ultimate steady state, right? But in a lot of scenarios, you want to be in a more realistic regime when you had like a set of species to which a given environment was exposed, right? And this is going to be a smaller subset of this big species pool. And the sampling basically takes account on that, because you can specify a number of species that you want to sample, and then you can specify the distribution under which you want to sample, meaning that like certain species will be selected more often, or like all species will be selected with equal probabilities or something like that, right?
Okay, it's a probability distribution for selecting species for special scenarios.
Yep.
Okay, thanks.
So the food parameter here allows you to take into account the following thing. Do you want to have this scenario with just one primary resource and then establishment of this whole trophic chain, or you want to externally supply like several resources, something like that? In principle, they also have default parameter for initial concentration of resource, but you can actually change it for your specific simulation. Regulation here takes into account how does your growth rate looks like in MacArthur setting. And I think they have a specific example about it, so we will come back to it later. So basically those two parameters, let's leave them untouched for now. So yeah, as you can see, a lot of things to specify. This is one of the sudden revelations you have when you start to work with those consumer resource models, is that like they are so simple, they're so intuitive. You can do them with pen and paper with some small scenarios, but then when you want to extend it for the complex community, you suddenly want to specify all those details. So basically, this is an example of how your initial state can look like under the sampling procedure. So each column here shows you a separate well, and white color shows whether you selected this species to be present in this well or not. Like black means you did not select the species. And for resource abundance, in their particular example, they just said that we provide R0, like first top resource at a high concentration, and then nothing else. All those other resources have to be produced by some of those species that will reside in the community. Basically, resource supply rates in their model can be of two different types. One is external, when you have some fixed amount of resource that you put in, and another is this self-renewing, which is a little bit more obscure assumption, I would say. So like this external one is the most realistic in majority of experiments. Basically for MacArthur model, they allow for several types of growth curves. And the difference between those growth curves goes as follows. Basically, type 1 is that linear growth that I was showing on my slides, where your growth of the species is defined by the sum of all lambda yields times resource. Type 2 takes into account carrying capacity. So it actually is of the form like divided by some capacity plus R here for each resource. And then type 3 is some Hill function that adds n in here. And it sometimes is used in some papers, but I would say that it's not a very popular thing. So type 1 is the most basic one, linear thing, like way to go assumption if you really don't have a strong opinion about this monod type carrying capacity kinetics. Okay. Yeah, here we're coming to metabolic matrices. Basically, those are the assumptions that are related to metabolic matrix that they use in the MacArthur model. So as I described, we basically want to specify how many species and how many resources we have. Then we want to specify whether we want to bias our models towards generalist or specialist, and provides some distribution for consumption and secretion. And here they basically show several examples of the matrices they typically use in their papers. One is that binary generation, when you have two classes and then this major class that they actually call wasted resources, because a lot of species can produce them, but almost nobody really consumes them. And basically, as you can see here, what those distributions change, they change like how pronounced are your growth rates for a certain class. Because if you use this Gaussian or binary consumer matrix, you will actually end up with a set of species that look like more towards the specialist side, because they have really strong growth rates for a small set of resources, and then they almost can't grow for many others. But like other assumptions, like a general one, this binary one or gamma distribution, can relax this constraint a little bit. So another very nice function that they provide in this package is that you can actually run a community dynamics for like one resource supply cycle and see how your microbial abundances are changing and how your resource abundances are changing. So as soon as you specified all those parameters and you sampled some community, you can actually see that some microbes come in, grow well on this top resource, but then they synthesize byproducts and they can actually go down in their abundances. But some other species can benefit from the byproducts and actually shoot up in this abundance plot. And yeah, this is all those nonlinear increases in byproducts and so on. And I think if I can actually run it real quick, you can see that those plots can look really differently depending on your sampling. Okay, let's wait a little bit. Does anyone have a question?
Yeah, there's a question in the chat, so maybe if we have some time. So
Okay.
Someone is asking, in these simulations, is the maximum amount of species that a well supports equal to the primary resource plus all the possible byproducts? Or are there more species possible?
I think yes, unless you put this enzyme budget constraint on your distribution.
Because then you expect that, that less or more?
It will be less than equal. Typically, it's really hard to get as many species as you had resources. Typically, you have slightly less, just because of this random sampling and random generation of parameters.
Yeah, but so you're saying now, if we add the constraint of enzyme budgeting, I expect even less?
No, if you add constraint on enzyme budgeting, you actually do normalization of your metabolic matrix.
Yeah.
You ensure that like the sum of your rho for each bacteria is the same, right?
Yeah.
In this case, when you do this solve of dynamic equations, you can actually have all those species surviving.
Yeah, exactly. Okay. Yeah.
Because none of them will be able to outcompete others because they all grow like simultaneously with an equal speed.
Yeah, then there's just a continuum of species.
Yeah.
So one thing that confused me a little is that when this person asked this question, I actually ran the code without this leakage. And I still get a lot of diversity. So I'm a bit confused about that.
Okay.
But maybe we could get into it once you're done.
Yeah, basically, Yeah, you see like different runs, different samples, and you can have like whatever plots here just because you picked different microbes from your pool. I think that if you set leakage to 0, you just have one species surviving. And this is like top competitor for the primary resource, right? And that's it.
I think I did something wrong. I'll check later.
And basically, when you start increasing it, this is where you start getting some other microbes here. And yeah, typically the higher is your leakage, the more interesting picture you may get. And like basically, typically from those abundances, you probably can have some idea about what is the rank of the byproduct each species relies on, because typically you have a loss of energy on each level, right, when you generate those byproducts. So like byproducts that reside on the bottom of this chain, they will be really scarce on energy and they can't really supply a lot of bacterial growth. So all those low abundant species, they stick in onto those really bottom byproducts. Okay, so
maybe unrelated question: is it possible to have some intrinsic noise here also, to have some variability between individuals or something like that?
Yeah, it's possible. I think. Right. Okay. In the MacArthur case, first of all, you have several noises. One is like your sampling procedure, right? Another is you have a distribution of growth and yield rates. So
as this is kind of variable parameters that you
yeah, I think
so yeah. But I mean, yeah, also, let's say little variations that we have maybe not accounted for, measurement errors or whatever, if we would want to simulate that.
I think they it's hidden somewhere in their code in those utilities, but when you generate initial state of your community, they have some intrinsic noise on the initial concentration of bacteria.
Yeah, I see. Okay.
Yeah, because this is what you want to have to account for experimental noise, right? Because in experiments, it's really hard to control like how many cells you put in each well. So yeah, they incorporated some noise on the number of cells you put in each of your well.
Isn't there also this thing on the budget, that the budget plus an error term?
Mm-hmm.
That you could also put noise there, I guess.
Yeah. All sorts of noises, whatever you want, is possible to put in here in those parameters. You're right. And then you can play and see how those dynamic trajectories will change. Okay. This part basically shows you several cycles of propagation and how does your ecosystem gets established, right? So what I show here, like top and bottom plot, is bacteria and resources respectively. So at first you have just your primary resource and whatever bacteria in very small quantities. Then you will start to accumulate byproducts a little bit due to this blue species here growing onto the primary resources and synthesizes those byproducts. And then you will have more and more byproducts, and the species dynamics will also be changed quite a lot. And then you will arrive at your final community state. And basically here in this example, they run the same set of species with this initial noise on sampling. So that's why you have those variation on those curves, because they all come in slightly different amounts. So like you have variability between your samples, but in the end of your dynamics, when you propagate it for many, many cycles, they will all be stabilized and they should arrive to one stable point with abundances which are kind of set by your gross kinetic parameters. Okay, so then I think they also provided one of the nice examples of how you can use this setup with wells to model some experimental setup. So they grabbed this paper which tried to explore the following scenario. In natural ecosystems, you sometimes have this succession, and in this particular paper, it's called like range expansion. And they studied the following thing. You have two species. One species can grow on some complex carbohydrate and output some byproducts. But then it can't really grow really well on those byproducts. It can utilize them a little bit, but not as good as other species that can come into this ecosystem and just thrive on those byproducts. So they did it in this well plate, and they showed that if you do the following thing, if you start with in a corner of your well plate with community of those two species, one is they call cooperator species that can degrade this primary resource, another is this other species that can benefit from this byproduct. Here in this part, those two species are established in some ecosystem, but then you can start migration events to nearby wells and see how they're propagated. So in like each propagation step of this experiment, when you pass your culture from old media to the fresh media, you also shift to one cell here, and then on the next step, you will shift your community to one step here, and so on. And then you can see that actually in these border cells, what happens is that you first have establishment of this primary degrader, and only in the next generation, other species will be able to come in and jump onto its byproducts. So this is what they actually model in this experiment. So they say that we grab like first half of our wells, and there we have just two species established, but then we start the passage into those empty cells. And this is basically how it happens. So this cooperator is this primary degrader strain, and you can see that it actually has this front that gets propagated throughout your experiment, and like the dark blue means that you have a high abundance of this degrader strain. So this is a like longest propagation, right? So this degrader lives on this end well, but here your byproduct consumer is already established, and it kind of kicks out this primary degrader. So I think it's kind of nice demonstration how you can explore different experimental setups in this well-played scenario using this model. And to the last thing, they also demonstrated how you can incorporate your own growth functions if you want, and like utilize it with their package. So here they use the Stillman model, for example, with Liebig's law as the main assumption for the growth rate. So what you need for that, you just have to define how your bacteria growth rate behaves. So here's this. This is the mean function. this is sum of resources multiplied by corresponding parameters. The same you do for resources. Then you can just utilize those growth functions to define dynamics, specify some dimensions. You will have to specify some additional parameters for those growth functions that you want to use, and they show it here. And basically, those are the cycles I was talking about, because here they demonstrate that if you specify 5 types of essential resources, you can have like all those wiggles here when you have some species coming in, dying out, other species are taken over. Your resource abundances will also behave wildly, and you will have to experiment a lot with your parameters to get rid of this unpredictable behavior, actually. And yeah, this is just a demonstration how this whole chaotic cycling looks like in the model. And I think it's more or less it. It just shows you how to play with this package. Whatever is left in this specific tutorial is just proofs that if you really don't want to run this whole dynamic integration of your differential equations and you're just interested in this endpoint, like who survives at what abundances and so on, you don't really need do this ODE solving. You just can use their optimized expectation maximization algorithms that will find this final state for you. And if you want to model a very complicated ecosystem with many species and like huge species pool, it's probably best option because it speeds up calculations a lot. And then you can just compile all these statistics of endpoints and start to explore like how does this steady state look like, what is the species prevalence in them, and so on. Like